Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my session. So yesterday we were done with this chapter, that is biomolecules. So today I want to discuss questions. I want to see how you're able to answer the questions and how many correct options are you able to give me and how many attempts you give me the right answer in. So that is also very important. Yes, Prince, a very happy good evening. So thank you so much for being on time. So please do like, share and subscribe. Yes, a very happy good evening. So shall we move into biomolecules? Yes. So I hope now biomolecules is pretty clear to everyone and there is no doubt. So we are getting stronger day by day with the concepts. So tomorrow I will start with cell cycle and cell division so that unit will be over. So in class 11 I'm yet to revise plant physiology and animal physiology. So sorry, plant and human physiology. So after cell cycle and cell division I'm planning to move to human physiology. So let's see, it depends on which one you ask me. So because recently we were done with plant physiology, so I thought we will start with human physiology after your cell cycle and cell division. But let's see, it's okay, it's not a hurry. So important questions from biomolecules, and this is my schedule for tomorrow. So tomorrow we are discussing PYQ of class 12. So we are discussing PYQ of class 12 from the same paper, that is 2020. So I will try to finish off 44 questions tomorrow in one arm so that it gets over quickly. Okay, so you want human physiology first? Okay, Prince. So after cell cycle and cell division, I'll start with your human physiology. Okay, yes. And tomorrow at 6 p.m. so we are meeting again. We are talking about a new chapter, that is your cell cycle and cell division. So today we'll be done with biomolecule once and for all, and then we'll move into your cell cycle and cell division. Okay, yes. Okay, fine. So this is my schedule. So we were here talking about biomolecule. Yes, Tarani, a very happy good evening. Yes, you need human physiology. Okay, I will go forward with that. Yes, thank you so much for joining my lecture. So please do like, share and subscribe. All right. So what were we discussing in biomolecules? So we have discussed a lot of things. So first we started with, okay, hi Shimpi. Yes, bonjour. So thank you so much for joining my session today. So please do like, share and subscribe, Shimpi. So we were speaking about a lot of compounds basically. So we were talking about two different fractions. So one is the acid soluble fraction. So acid soluble is your filtrate. And then acid insoluble is your retentate. So this includes amino acids, this includes your lipids, this also includes your nitrogenous bases, and these are basically called as your biomolecules. Okay, yes. Previous lectures, so go as per your wish. Okay, so that's okay. So let's see, it depends according to the situation in the last minute. So how many of you demand human physiology? Otherwise we will go with plant physiology. Yes, Ayyappan Shaktivel, a very happy good evening. So thank you so much for joining my session. Yes, I'm good, thank you. How are you? So please do like, share and subscribe the session, please. So this is retentate. So this includes your polypeptides, polysaccharides, and your nucleic acids, and to some extent lipids, and these are called as biomacromolecules. Right, so we were talking in detail about these things, so I'm not going in detail again. Hi Sweta, yes, a very happy good evening. So how are you? So thank you so much for joining my session today. So please do like, share and subscribe. Okay, then the important part of this chapter is your enzymes. So enzymes are basically proteins. So how does an enzyme work? Yes, I'm good, Sweta. Thank you. How are you? Okay, how does enzyme work? So enzyme has an active site. So your substrate binds to the enzyme. Yes, and then it forms a transient enzyme substrate complex, which very quickly becomes your enzyme product complex, and thus your enzyme and your products are released. So this is how your enzyme works, right? Okay, so here, so what are the factors that affects enzyme action? So first one is your temperature, your pH, Then you have what is known as substrate concentration. So what is this substrate concentration? So number of enzymes and the number of substrate. So if number of enzymes are, for example, 50, if number of substrate is 60, then as the concentration of substrate increases, then the velocity of the enzymatic reaction also increases. So when the substrate concentration increases, the velocity reaches maximum. So that is known as Vmax. So the substrate concentration needed to reach half Vmax, half maximal velocity, is known as your Km value. So I hope you remember. Then you have one term, what is known as activation energy, which we represent it like this. So activation energy is the energy present in your substrate in its ground state to that present in your transition state. So in an enzyme catalyzed reaction, your activation energy is less. So this is the correlation. So this is what we were discussing in yesterday's class. Okay. Yes, Swami, a very, very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Come on guys, don't forget to do that. Okay. So here I want to teach you one more thing. So yesterday we were talking about what is known as inhibition in your enzyme. And I taught about what is competitive inhibition. So what is competitive inhibition? So when I have a substrate for a particular enzyme, so there is another molecule which is also very similar to your substrate. So your enzyme has, or this substrate has more in number, so this quickly binds to the enzyme, but ideally this should have bound. But since if this binds, then the enzymatic activity will not proceed forward. Hi Anthony Abhi, yes, a very, very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. Right, so this molecule is known as an inhibitor and it is known as a competitive inhibitor. Right, so one example of this competitive inhibitor, so this inhibitor is your malonate. This is an example, but the actual substrate is your succinate. The enzyme is your succinate dehydrogenase. So your malonate and succinate are very similar in structure, but ideally your succinate should bind to the enzyme. But since malonate is more in concentration, it binds to the active site of the enzyme, thus inhibiting the reaction. So we know an enzyme is very specific to the substrate. Though from the structure it can go and bind, but the enzymatic activity will not proceed. So if two tablets are similar, so one is for diarrhea, one is for paracetamol, both look similar. So if you take diarrhea, your fever will not, a diarrhea tablet, your fever will not get cured, right? So though this binds to the active site of the enzyme, so the enzymatic reaction will not proceed. All right, clear everyone? So this is an example that I want to give you. So moving further, there's another type of inhibition known as allosteric inhibition, or feedback inhibition, or non-competitive inhibition. So what is this? Allosteric feedback non-competitive inhibition. So if A is giving me B in the presence of an enzyme E1, so now this end product itself inhibits the activity of the enzyme to stop this production. Suppose if A is giving me B in the presence of an enzyme E1, and if the concentration of B is very high, the cell does not want so much of B, this product itself will inhibit the particular enzyme. So that is known as allosteric feedback or non-competitive inhibition. Okay, yes. Okay, so yes, hi Antonia B. Fine, clear? Now, one example for this, you have fructose 6-phosphate in your glycolysis, right? So this is converted into fructose 1,6-bisphosphate. And here the enzyme is phosphofructokinase. So, if there is more amount of fructose 1,6-bisphosphate, this itself will inhibit the activity of this enzyme, thus stopping this process. So, this is an example of allosteric feedback or non-competitive inhibition. So, these are the two different types of inhibition. Is this clear to everyone? Yes, any doubts? Should I explain once again? Yes, quick, let me know. Come on, guys. Yes, quick. So, everyone understood this? Yes? No doubts? Yes, it's clear to everyone? So, you understood what is competitive inhibition, what is non-competitive, allosteric or feedback inhibition? Yes? This is clear? So, shall I move forward? So, this is the only bit that I didn't teach properly yesterday. So, I hope this is clear. Yes. So, now you remember what is cofactors? The non-protein part of your enzyme. Right. So, here you have three types. One is prosthetic groups. The other one is coenzymes. And the last one is metal ions. So, prosthetic groups are tightly attached to the protein part of your enzyme. So, that is prosthetic group. Coenzyme is when it is transiently attached to the protein part. And the metal ion, which forms coordination bond with the enzyme as well as with the substrate. So, that is your metal ion. Yes? Clear, everyone? So, shall I move forward? Yes? Okay, so after this, I have a set of 10 questions. So, each one of you will evaluate yourself and let me know how much you have scored out of 20 at the end of the session. Okay, yes. So, in how many attempts you give me the right answer, so that also matters. So, for every question, I will give you 1 whole minute time. So, look into the question and then give me the answer. Yes, okay. Can you say the definition for allosteric inhibition? Yes. So, the definition for allosteric inhibition is when the end product of a particular reaction inhibits the enzyme catalyzing that particular reaction. When the end product of a particular reaction inhibits the enzyme catalyzing that particular reaction, it is known as allosteric inhibition. Okay, Prince, is this clear? Yes. Yes, any other doubts? So, please feel free to ask me. So, this is the last division of syllabus wrap up that I am going slow. So, if you have your exam on 1st of August, so please take all the sessions seriously. Otherwise, it will be tough. So, now I remember. So, I want to tell you. So, today or this century marks almost, so this year almost marks 100 years for the discovery of your insulin. So, what is insulin? So, it is known as centenary year. So, it is a centenary celebration. So, this year marks 100 years for after the discovery of insulin. So, your insulin was discovered in 1921. 1921 to 1922. So, this year marks 100 years after the discovery of insulin. So, insulin was discovered by Frederick Banting. So, he was the scientist who was working on what led to diabetes mellitus. So, people were, what led to diabetes? So, diabetes has been a very, very serious or a grave disease back since then. So, they were trying to find out what causes diabetes. So, they were working on pancreatic structures, juices, and then they found out islets of Langerhans, and then is when the discovery of insulin happened. So, insulin is not a cure to diabetes, it is only a treatment for diabetes, and he won the Nobel Prize for this discovery. So, it was a very breakthrough discovery, right? So, without insulin, what will we do now? So, insulin was the first protein which was sequenced. Insulin was the first chemically synthesized protein. Insulin was the first recombinant protein which was produced in bacteria. So, this year marks 100 years after the discovery of insulin, or the 100th year for the discovery of insulin. So, insulin was discovered by Frederick Banting. So, he discovered it in 1922. So, insulin is a treatment to diabetes. It is not a cure, and this person won Nobel Prize for the same. So, this was the news in Nature today. So, it was a very beautiful article. So, it talked about the life story, how the experiment started, how Frederick Banting discovered insulin. So if you are more interested, so please go forward and Google this. So insulin is a peptide hormone secreted by the endocrine nature of your pancreas. So insulin decreases the blood glucose level. It is secreted by the beta cells of your islets of Langerhans. Yes, ma'am, a very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. Yes, insulin is the first protein to be sequenced, first chemically synthesized protein, and the first protein to be used as recombinant protein in your bacteria. All right, yes. Okay, so moving further. So question number 1. So please evaluate yourself. So I have totally 20 questions. So in how many attempts you give me the right answer? So that also matters. Okay, so the turnover number of enzyme depends on the size of enzyme molecule, the number of active sites, concentration of the substrate molecule, molecular weight of an enzyme. Yes. So let's see how many of you give me the right answer. So guys, please do like, share and subscribe. Yes, turnover number of enzyme depends on. Shimpi, option D is wrong. So turnover is ability, so turnover is how much of product it produces. D is the wrong answer. I am not asking turnover number of biomolecules, I am asking turnover number of enzymes. So Shimpi and Prince has given me 2 attempts, but still it is the wrong answer. So you will consider it as wrong. Okay, Shimpi and Prince. The right answer is the turnover number of enzyme depends upon the number of active sites. Depends upon the number of active site. Yes, Anthony, you're absolutely right. So if this is an enzyme and if the enzyme is going to give me product, how many active site it has? Sometimes it can have more than 1. Right? So depending upon the active site, the turnover molecule of the enzyme varies. So question number 2. The Km value of the enzyme is the value of the substrate concentration at which the reaction reaches to 0, twice Vmax, half Vmax, 1/4 of Vmax. So Km is the value of the concentration of the substrate. Km value of the enzyme is the concentration of the substrate that is required to reach half maximal velocity. Yes, absolutely right. Okay, so everyone have got it right. Good. So question number 3, feedback inhibition of enzyme is affected by enzyme, substrate, end product, intermediate end product. Yes, Anthony, you're right. Enzyme, substrate, end product, intermediate end product. Yes, what is the answer? So Shimpi, just now I explained what is feedback inhibition. So what did I say? I said when A is giving B and if the concentration of B is too high, so B itself will inhibit this enzyme. So the end product is affecting the enzyme. So here it is option 3. Yes, absolutely right. So Anthony has got all the 3 right. Okay. So question 4. So which one of the following statement is correct with reference to enzyme? Holoenzyme is equal to apoenzyme plus coenzyme. Coenzyme is equal to apoenzyme plus holoenzyme. Holoenzyme is equal to coenzyme plus cofactor. Apoenzyme is equal to holoenzyme plus coenzyme. Yes. Yes, absolutely right. All of you have given me the right answer. So holoenzyme is the whole enzyme. It is equal to the apoenzyme, that is the protein part, plus the non-protein part. Right? So this is the right answer. Yes, absolutely right, guys. So moving forward, question number 5. Relation between amino acid and protein is similar to the one found between glucose and fructose, thymine and uracil, nucleosides and nucleic acid, nucleotides and nucleic acid. There is no hurry. Please take your time to answer. So there is no hurry. So amino acid and protein. So proteins contain your amino acid. Yes. What is the similar one? So similar relation is shared by what? Yes, quick. So nucleosides and nucleic acid, is it right? So nucleic acids are made up of nucleosides. Is that so? So Prince, you say nucleosides. So nucleosides are only your nitrogenous base plus your sugar. But a phosphate group is also needed. So this forms your nucleic acid, right? So 3 is the wrong answer. It is nucleotides and nucleic acids. So nucleic acids are polymer of nucleotides. So proteins are polymers of amino acids. So similarly, nucleic acids are polymer of your nucleotides. So here the right answer is option D. Yes? So out of 5 questions, what is your score, everyone? Yes? Yes, Anthony, you're right. So 5 is D. Yes, you're absolutely right. Yes, everyone. So shall I move forward? Okay, so question number 6. Antiparallel strands of DNA molecule mean the phosphate groups of 2 DNA strands at their ends share the same position. The phosphate groups at the start of 2 DNA strands are in opposite position. One strand runs clockwise, one strand runs anti-clockwise. So Prince has got 3 out of 5. Shimpi has got 2 out of 5. Okay, yes. Guys, please do like the session. Yes, question number 6, what is the answer? Question number 6, what is the answer? Vaibhav, Rana, hello, hi. So thank you for joining my session. So please do like, share and subscribe. Yes. So Prince says B, Shimpi says B. Yes, absolutely right answer. The phosphate groups at the start of 2 DNA strands are in opposite direction. So 5 prime, 3 prime. So 5 prime you have phosphate group. So 3 prime you have OH. So the other strand, so it is 3 prime to 5 prime. So the phosphate has a different position. So option B is the right answer. So question number 7. All enzymes are proteins. This statement is now modified because an apparent exception to this biological truth is aryl sulfatase, dehydrogenase, ribozyme, nitroreductase. So what is the exception to this line? All enzymes are proteins. Yes, Prince, you're right. Okay, others? Yes, others? Yes, Shimpi. Yes, Antonia B. What is the answer? Yes, what is the answer? Ribozyme, because your RNA also acts as a catalyst. So this is the right answer. Yes, okay. So good. So Shimpi and Antonia B, I hope you agree. Yes. So question number 8. Select the type of enzyme involved in the following reaction. So dehydrogenase. Ma'am, why your English accent is so different than us? So do I speak differently? So do I don't have the same accent as you? Maybe because my English is a bit fluent, that's all. Yes, so S, so this is one substrate, this is the other substrate. So it has a group G attached. So here the group G is shifted between the substrate. So what is the answer? Yes, it is transferase. So this helps or catalyzes the transfer of groups between substrates. So transfer of groups between substrates. Yes, absolutely right. Very good. So question number 9. So transition state structure of the substrate formed during an enzymatic reaction is permanent but unstable, transient but unstable, permanent and stable, transient but stable. So transition state of the structure of the substrate formed during an enzymatic reaction. So this is your transient state reaction. So this is the complex. So this is known as your enzyme substrate complex. Yes, why? So Shimpi says permanent and stable. Is this structure permanent? I am saying transient. So transient means it is short-lived. So the meaning of the term transient is short-lived. So it is transient and it is unstable. It quickly gets converted into enzyme product complex, right? So the right answer is transient and unstable. Yes, option B is the right answer. Yes, very good. So moving forward, question number 10. So for a protein to have a quaternary structure, it must have 4 amino acids. It must consist of 2 or more polypeptide subunits, consist of 4 polypeptide subunits, have at least 4 disulfide bridges. Yes, question number 10. Yes. So quaternary structure of protein is seen in your hemoglobin. Yes, because it has 2 alpha subunits and it has 2 beta subunits, right? So it consists of 4 polypeptide chains. So then it becomes a quaternary structure. So here they are asking for a protein to have a quaternary structure, what is the rule? The rule is that it should consist of 2 or more polypeptides, need not be 4. It can have 3 as well. But the example that I gave you is 4. Okay, the ideal thing is that it should have 2 or more polypeptide subunits. So the right answer is option B. No, Antonia B, it is not D, it is B. Okay, yes, fine. So question number 11. So the quaternary structure of the human hemoglobin is best described as a dimer of identical subunits, dimer of different subunits, tetramer of 4 different subunits, or tetramer of 2 different subunits. Disulfide bond in DNA. So do you have disulfide bond in DNA? So DNA does not have sulfur. DNA has only phosphorus. So proteins have your sulfur and DNA does not have your sulfur. So there is no disulfide bond in your DNA. Okay, Antonia B? Yes. So question number 11, the quaternary structure of human hemoglobin is best described as? Yes, question number 11. Tetramer. Okay, so they're directly asking about hemoglobin. So this has 2 alpha subunits and 2 beta subunits. So together it has 4 subunits. So it is a tetramer of 2 different subunits. So here the right answer is option D. So tetramer of 2 different subunits. Okay. Ma'am, in DNA, B bond have? No, I don't understand. So in DNA, where do you have disulfide bond? It is hydrogen bond here. Where do you have disulfide bond? Yes, where do you have disulfide bond? Hi, sweetie. Yes, a very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. Yes, Antonia B, what is your confusion? So where did sulfide bond come in DNA? Yes. Yes, it is seen in your proteins, your immunoglobulins, your antibodies are proteins. So proteins have disulfide bonds, I agree, but not your DNA. No, Anthony, there is no disulfide bond in your DNA. Okay, yes, so tetramer of 2 different subunits. So question number 12, at temperature near freezing point, the enzymes are inactivated, activated, slightly inactivated. Yes, so please think, take your time and then give me the answer. Yes, quick. Yes, what is the answer? So at temperature near freezing point, the enzymes are inactivated because you have an optimum temperature at which your enzyme works best. So this is your optimum temperature, right? So A chain, B chain comes in your insulin. So insulin is not here in your DNA. Insulin is a peptide. It is a protein hormone. Okay, Antonia B? Yes. So here it is option A, that is inactivated. Yes, right. So question number 13. So hexokinase, so that is used to catalyze the reaction glucose to glucose 6-phosphate, belongs to the category transferase, lyases, oxidoreductases, isomerases. Yes, question number 13, what is the answer? Question number 13, what is the answer? Yes. Question number 13. Yes, Shubham sir. Hello, hi. Very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. 13 is D. Is it isomerase? Antonia B, when it moves from glucose 6-phosphate to fructose 6-phosphate, it is isomerase. Otherwise, it is not isomerase. So here the question is glucose to glucose 6-phosphate. So here it is transferase. It is transferring phosphate from your ATP. So that is why this becomes adenosine diphosphate. So here you have triphosphate. Here it becomes diphosphate because a phosphate group is getting transferred to glucose, right? So it is transferase. All right? Yes? Okay. So question number 14. What will happen when the cofactor is removed from the enzyme? So cofactor is the non-protein part. Catalytic activity of the enzyme is lost. Enzyme preserves in a temporarily inactive state. The substrate molecules are not closely related to enzyme molecule, both B and C. Yes, what is the answer? Yes, what is the answer here? Yes. So Shimpi says D, both B and C. Yes, is that your answer, Shimpi? No, if a cofactor is removed from an enzyme, it is directly the catalytic activity of the enzyme will be lost because cofactors also help in the complete enzyme activity. So if a particular cofactor is removed from an enzyme, the catalytic activity of the enzyme will be completely lost. Okay, so it is option A. Fine? Okay, so question number 15. So primary structure of polypeptide is stabilized or secondary structure of polypeptide is maintained by disulfide bonds, ionic bonds, hydrogen bonds, hydrophobic interactions. Primary structure of the polypeptide is stabilized by. So primary structure is like this. So this structure is stabilized by, or the secondary structure of polypeptide is maintained by. So secondary structure is like this. So this is your alpha helix and this is your beta pleats. So they are stabilized or maintained by what? So Shimpi says ionic bonds. So I guess Shimpi has some idea about chemistry. So she's saying ionic bonds. But no, that is the wrong answer, Shimpi. Yes, what is the right answer? Yes, guys, what is the right answer? Come on, quick, what is the right answer? Others, please try. Yes, so Shimpi is smiling. Yes, what is the right answer? Disulfide bonds. Okay. No. So they are stabilized or they are maintained by hydrogen bonds. So the stability is given by hydrogen bonds. Okay. Yes. Clear? So moving forward to question number 16. So the catalytic efficiency. Okay. So Prince says hydrophobic interaction. No, Prince. So it is hydrogen bonds. Yes, Swami, you're absolutely right. Very good. So question number 16. The catalytic efficiency of two different enzymes can be compared by Km value, pH optimum value, formation of the product, molecular size of the enzyme. Yes, come on, quick. What is the answer? What do you think will be the answer? Yes, quick. The catalytic efficiency of two different enzymes can be compared by the Km value. That is absolutely the right answer. Very good, Prince. Very good, Antonia B. Good. So, Shimpi, what is your guess? What is your guess? So you agree with the Km value? Yes. Okay, go Swami. So question number 17. Enzyme that breaks nucleic acid into nucleotides belong to which of the following class? So oxidoreductases, transferases, hydrolases, lyases. So enzymes that break nucleic acids into nucleotide belongs to which of the following classes? Oxidases, transferases, hydrolases, lyases. Yes. So the enzyme which breaks nucleic acids, they are your nucleases. So this belongs to a broader category, that is your hydrolases. This causes the hydrolysis of a particular bond. So this is option C. Yes, this is very close by, but this belongs to a family known as hydrolases because it causes the breaking of a particular bond. So two nucleotides are linked together by a phosphodiester bond. So if I am breaking nucleic acids only into nucleotides, so this is how my nucleic acids are. So I am just breaking this into individual nucleotides. So this is nucleases. But if I want to break this into nitrogenous base plus sugar plus phosphate, then it is lyases. Okay, yes. So you are Asad's student, right? So that is the reason. Okay, Shimpi? Yes. So question number 18. When triglycerides are solid at ordinary room temperature, they are called as oils, gases, fats, none of these. When triglycerides are solid at ordinary room temperature, they are called oils, gases, fats, none of these. Yes, absolutely right. So this is fats. So if they are solid at room temperature, they are called as fats. Yes, very good. So Shimpi, you have also given me the right answer. Very good. So question number 19. So when glucose is degraded to lactic acid in skeletal muscle, energy is liberated. So which of the following conclusion is correctly stated with the above statement or the enzyme? So it is the catabolic pathway which releases energy. It is anabolic pathway which releases energy. The energy liberated during this degradation is trapped and stored in the form of chemical bonds or none of the above. Yes, what is the right answer here? So question number 19, last but one question. Yes, what is the right answer here? Question number 19. Yes, guys, come on. So question number 19. So the clue is given in the question itself. So energy is liberated. Only in one reaction energy is liberated. So your metabolic reactions are of two types. One is catabolic, one is anabolic, right? So catabolic is like climbing down the hill. So here you have larger molecules which breaks down to form smaller molecules. So energy is released. So anabolic is climbing up the stairs. So from smaller molecules you have larger molecules formed. So here the question itself says energy is liberated. So it is the catabolic pathway which releases energy. Okay, yes. So the last question itself. So question number 20. The stored form of sugar in animals is a homopolysaccharide, heteropolysaccharide, oligosaccharide, disaccharide. So homopolysaccharide is you have repeatedly same sugars present but n number of sugars. So n number of sugars but different sugars, glucose, fructose, etc. So oligosaccharide is less than 10 sugars that are present, it is oligo. So di is 2 sugars. So the stored form of sugar in animal is homopolysaccharide because the stored form of sugar is your glycogen. So glycogen is made up of repeating structures of glucose. So it is the same. So it is homopolysaccharide. Alright, is this clear? Yes? Done? So are we thorough with biomolecules? Yes, now give me your scores. So please self-evaluate yourself. So what is your score now? Yes, let me know your scores. Yes, guys, come on. So Prince, Anthony, Shimpi, Swami, what are your scores? 13 by 20. Okay, Prince. So please solve more questions in biomolecules. So Anthony, what is your score? Yes, Prince says he's clear. Okay. So ma'am, Anthony says, ma'am, 17 out of 20. That's a pretty brilliant score. Okay, fine. All right, good. So not bad. So please study your NCERT. So go through. If you have any doubts, ask me in the next session. Okay? Yes? Fine. So this is my schedule tomorrow. So tomorrow we are discussing PYQ of the year 2020. That is also fine, Swami. So please practice more questions. You will come to perfection very soon. Okay. So PYQ of 2020. So we are discussing class 12 topics tomorrow. And we are starting a new chapter, that is cell cycle and cell division. So please join me for the same. So tomorrow it is lecture 1. Okay. Yes. So after cell cycle and cell division, so should I start human physiology or animal physiology? So please write your opinions or suggestions in the comment section. So I will go through it. All right. So thank you so much for joining my session. And if you genuinely liked my way of teaching, so please do like, share and subscribe. This is me wishing you more and most. So stay tuned and take care. I will meet you tomorrow at 4 p.m. So until then, bye. Okay, Prince, I will start with human physiology. All right, guys, bye.